Okay, so the learning outcome for today's lesson is to describe the fluid mosaic model of the membrane structure and to describe the arrangement of cholesterol, glycolipids, proteins, and glycoproteins in the cell surface membrane, and to also describe the roles of these molecules in the cell surface membrane. And the fourth one, we will touch on it in the next lesson because it needs more explanation on this part. And this is new to you, okay? So overall, a membrane, right? What are the membrane that you have learned so far from chapter 1, Charmaine? Is my Charmaine here? No? Okay, Januli?
Cell surface membrane.
Some more?
I'm not sure.
Inside the cell, other than cell surface membrane, are there any other membranes? Generally,
The membrane surrounding the organelles.
yes, the membrane surrounding the organelles like nucleus, rough ER, smooth ER, right? And then the lysosome, Golgi body, Golgi apparatus, secretory vesicles, right? So all these membranes are part of the membrane system of the cell, right? Other than that, we also have got membrane surrounding the mitochondria and also the chloroplast. So all of them are called the plasma membrane. Okay, plasma stands for liquid. So that means membrane is actually liquid, okay? And then the membrane that is surrounding the outer boundary of the cell is called the cell surface membrane. Okay, so therefore, when you label the outer layer of the membrane of the cell, it is more accurate to label it as cell surface membrane rather than plasma membrane. Okay, so thank you, Januli. So membrane is made up of all these biological molecules that you have learned. So it includes lipids, right, proteins and carbohydrates. So for lipids, there are a few types. One is a phospholipid, another one is cholesterol. Okay, you'll learn more later on. So this is the basic structure of a phospholipid molecule that you've learned in chapter 2. So very importantly, what you have learned is firstly, it is made up of a glycerol with 2 fatty acids attached to it. Okay, and then the third carbon is attached to a phosphate group. PO4 is phosphate group. And to the phosphate group, there's another group called choline. Okay, choline, please learn up the word choline, which is made up of a nitrogenous compound that is positively charged. So both, you can see there are 2 locations here that is charged. Can you find the 2 locations that is charged? Hoshini?
The oxygen.
Yes, this oxygen of the phosphate is negatively charged. Yes, very good. One more.
And plus the positively charged delta.
Yeah, the N plus of choline. The N plus of choline is positively charged. So you can see that in this phospholipid molecule, there's this region that contains charge. So the region that's contained charge, is it water soluble or not? Samir?
Yes.
So this entire, right, this choline plus phosphate, right, this whole group here, okay, you see the nitrogen and phosphorus, they are, okay. So this choline and phosphate, both are charged. So this is the hydrophilic head of the phospholipid molecule, the hydrophilic head. So this is the head of the molecule, okay? And of course, you've got the shoulder of the molecule, which usually you don't show them. So the shoulder is actually the glycerol, right? The shoulder, right? This part here. So usually we don't show them. Then in a short form of the molecule, then you got the tail, right? The two tails, the fatty acid tails that are hydrocarbon tail. So this hydrocarbon tail is hydrophobic. So this is what you have learned about this molecule, which is the basic molecule that make up the membrane. So you need to know the structure of this molecule quite well, all right? So when you say this molecule can be divided into two parts, the hydrophilic head is the phosphate plus the choline. Then if you say hydrophobic tail, so the tail you are referring to the fatty acid tail. So that means we are ignoring the glycerol, which is the shoulder already, okay? So we're not talking about the glycerol anymore. So you need to know very accurately what those terms mean, okay? Which part belongs to which term. Okay, so let's look at what happened to this phospholipid molecule. All right, so you need to draw this into your notes. So this is a symbol that we use to represent the phospholipid molecule. So the blue color circle represents one polar phosphate head, okay? And this is hydrophilic. Hydrophilic means water-loving. And because it's water-loving, it will arrange facing the water. Then there are two red color lines here, which represent the two non-polar fatty acid tails. So because they are hydrocarbon, so carbon made with hydrogen attached to it, so hydrocarbon. So they are hydrophobic, means water hating. Phobia means they don't like. So therefore, in water, they definitely try to avoid the water. So they try to face away from the water as much as possible. So when you throw the phospholipid molecule into the water, you expect the phosphate head to be facing the water because they love water. Then the fatty acid tail will try to face away from water because they don't like water. So it's like somebody that you like, you want to see them, right? You want to face them. Somebody you don't like, you want to hide away, you don't want to see them, right? So it's the same thing. So when you throw these molecules into the water, this is how they will arrange. You can see that, not? In water, fatty acid tails arrange facing each other. They don't want to face the water. So this is one of the ways that they will arrange themselves when you throw them into the water. But there are other ways that they will also be arranging themselves in water. So let us look at that, okay? Now, arrangement of phospholipid molecules in water, you need to draw this into your notes. A ball-like structure, which is called micelle, okay, micelle. Sometimes when you read the word, right, if you do not know how to pronounce it, please go to Google and search it, okay? Search the meaning of the word, and then usually they will actually pronounce it for you, right? Or else you start to pronounce it micelle, okay? Micelle or micelle, whatever. So it's called micelle, okay, micelle. So A is ball-like structure, micelle. B is sheet-like structure, the phospholipid bilayer. So let us look at the structure which you need to draw. So you can see that all the fatty acid tails, right, they are facing each other away from water, and the phosphate head are facing water. So they are at the best position. Those that like water face water. Those that don't like water will not be facing the water. So this is the ball-like structure, micelle. Second, if you've got more phospholipid molecules, they can start to arrange into sheet form, okay? Sheet form is like a flat sheet of paper. So made up of phospholipid bilayer, means the phospholipid molecules arranged in two layers, right? This is one layer, okay? This is in the center, right? So this is the second layer. So the top layer and the bottom layer, okay? So bilayer, phospholipid bilayer. So again, the red color fatty acid tails, they are not facing the water at all. So they are protected away from the water by the phosphate head. Okay, so draw these two into your notes. Then, oh no, you cannot see the words, can you? Okay, there's some error that I need to, some setting I need to modify. Bring to the back so you can see. Okay, so let's do it again. So phospholipid bilayers forms the basic structure of all the membranes of the cell, right, and inside the cell. And okay, so there's another arrangement of phospholipid molecule in water-air interface. So in water-air interface, what do you think the phospholipid molecule will look like? Okay, so it's very, very interesting. So it look like this. The head will be immersed in water, but the two tails will be pointing towards the sky. You know, this is called water gymnastic, right? Okay, water gymnastic. The whole body is inside water, but the leg is outside the water, okay? Water gymnast. Right, so this is the arrangement in the water-air interface. So you can also draw this into your notes, okay? So putting everything together, when you got a lot of phospholipid molecules where the hydrophilic polar head, you see, when you say the polar head, it includes choline and phosphate. When you say the phosphate head, you are referring to the phosphate only, okay? So be careful what is the word that you use to name them. Polar head is this whole thing, right? Whereas phosphate head is only the phosphate, this one only, okay? So you got the hydrophilic part and the hydrophobic part. So glycerol is the hydrophobic part, okay? And the tail are the two red color one. So when you put them in water, straight away they'll form the phospholipid bilayer, all right? So hydrophilic head orient towards the water, hydrophobic tails orient away from the water. Orient means how they arrange themselves, okay, orientation. So phospholipid bilayer is fluid. Fluid means like water. Now, when you talk about water in the form of liquid, now it is liquid because water molecules are free to move, isn't it? They're free to move. So when phospholipid bilayer is fluid, that means the phospholipid molecules are also free to move. Okay, they're free to move. So individual phospholipid molecules is free to move laterally. That means they will only move sideways. Okay, laterally, please learn up this word, meaning sideways. Okay, so they can move to the left side, they can move to the right side. They only move sideways because the fatty acid tail, they don't like the water. So the phospholipid molecules cannot move up and down because when they move up and down, the fatty acid tail will be exposed to water. So they do not want to be exposed to water. So the only way is you move sideways, okay? You move sideways. So this is how the phospholipid molecules will move, and therefore the phospholipid bilayer is a fluid, okay? So it is like the shell of a soap bubble. You know, when you play, you blow the soap liquid, okay, through a spherical frame, then you get a lot of the soap bubbles, right? Okay, so the soap bubbles are actually liquid, isn't it? And they're actually moving. So the membrane is like the shell of the soap bubble. So you can imagine like this, but the viscosity of this fluid is like olive oil. Now, if you pour olive oil is quite viscous. Viscous means very thick. It's not so watery. So it's quite thick, okay? So our membrane, even though it's liquid, but it's quite viscous, okay? It's slow to move. It's sticky, okay? So the fluidity or the viscosity depends on the hydrophobic interactions of the fatty acid tail. So the more sticky, you see, water is sticky because of the hydrogen bonds between water molecules, right? Causing them to be sticky. Now, phospholipid bilayer, okay, the fluidity or the viscosity is how closely the phospholipid molecule can stick to each other. When they stick to each other more, they move less. So they become more viscous. When they stick to each other less, they will move more. So when they move more, the membrane becomes more liquid, more fluid. Okay, so whether they will stick more or stick less, it depends on the hydrophobic interactions of the fatty acid tails. Okay, so the more the hydrophobic interactions between the tail, that means they stick to each other more, so they tend to less free to move. So the membrane become more viscous. Okay, so let us look at this relationship of the fatty acid tail. So remember, when you learn about the fatty acid tail, the length of the tail can be variable. That means the number of carbon can be different. The number of double bond or the unsaturated bond can be different also. Okay, so on the left side, we've got 3 saturated fatty acid tails. Okay, and on the right side, we've got 3 fatty acid. One of them is unsaturated. So you can also know that the unsaturated part is actually this part here, where there's a double bond between these two carbon, causing a kink to happen at this location. Okay, remember I asked you to learn up the word kink. Please learn up, okay? Kink. So you can see that when the phospholipid, the fatty acid tail is saturated, the phospholipid molecules will be highly ordered packing. So they arrange in a manner that's highly ordered packing, okay? Whereas when we have got any fatty acid tail has got double bond, then the double bond disrupt the close packing of the fatty acid tail. So they cannot pack very close to each other anymore. So these 3 phospholipid molecules, they cannot pack very close to each other. So which means to say that only this part of the neighboring fatty acid tails can form hydrophobic interaction. Only this part can form hydrophobic interaction, and maybe this part only. Can you see that? Whereas for these 3, the entire length of fatty acid tail can form hydrophobic interaction. So you can see that hydrophobic interactions for the phospholipid molecules on the left side will be actually more than the phospholipid molecules on the right side. Can you see that? So which means to say, which molecule is harder to move? Curti, left or right? Which phospholipid molecules are harder to move, left or right?
Left.
Left side, right? Because the entire chain, all the entire chain is held together by the hydrophobic interaction. So they are attracted to each other, so they don't want to separate. So more difficult for individual molecules to move. Okay. So which membrane will be more viscous? Hui Min, left or right?
Left side.
Okay, so the right side will be more fluid, right? Which one will be easily frozen at lower temperature? Joyce.
Left.
Very good. So which cell containing this type of membrane will easily break in freezing? Will easily break and die? Samir.
I don't really understand the question. Can you repeat that again?
Okay, so as the temperature drops, right? As the temperature drops, which cell will die first? Okay, which membrane will freeze first? As the temperature drops, which membrane will freeze first? Left or right?
Left.
Left, correct. Freezing is when all the molecules form maximum bonding with each other, right? So of course the left one will freeze first. So if the left one freeze first, you imagine your cell, the membrane frozen versus the membrane is fluid. Which one is easily broken? Pei?
Left.
Left, right? So we don't want the membrane to freeze at all, because when the membrane is frozen, then you just have to push it a bit. The membrane will break and the cell will die. So we try as much as possible not to allow the membrane to freeze, which means to say we do not allow our phospholipid molecules to align with each other so closely. So we will not allow this to happen. So the double bond sometimes is quite good in the sense that it prevents the orderly packing, so that it will minimize the hydrophobic interactions, so that the membrane will not freeze at low temperature. So that's very important. Okay. So on the left side, there's more hydrophobic interaction, so membrane is more viscous. On the right side, there's less hydrophobic interaction, so membrane is more fluid. Okay, membrane cannot be too viscous, nor can it be too fluid also. Okay, it cannot be either way. Too much or too little also no good. So both extreme is not acceptable for membrane. So how do we maintain the fluidity of membrane? Not too fluid, not too viscous. We use another molecule to do that. Okay, so we'll learn that one afterwards. So now we're going to learn the fluid mosaic model of a membrane structure. So this is a fluid mosaic model of membrane structure in 3-dimensional structure. Okay, so you need to be able to explain what is fluid, what is mosaic very accurately. Fluid, you are referring to the phospholipid molecules, which are free to move laterally. Okay, and also don't forget the protein molecules are also free to move laterally, because when you remember, when water molecules move, it carry with it whatever that dissolve in it. So when phospholipid molecules move, whatever that stay within the bilayer can also move with it. So it's also fluid. So the protein inside it is also fluid. Okay. Now, what about mosaic? So fluid is referring to the phospholipid molecules mainly can move laterally, but the proteins also can move. Mosaic is referring to the protein molecules in the membrane, where the protein molecules are scattered and float freely in the phospholipid bilayer. So this is the meaning of mosaic, means referring to protein molecules are scattered around the phospholipid bilayer. So therefore, when you look at the protein from the surface, you can see that the pattern of the membrane is like a mosaic. The tile, mosaic tile, I don't know whether you know what's mosaic tile or not. Mosaic tile is a tile like this with a lot of dots on it. Okay, irregular dots and big and small irregular dots. Can see, cannot see. Okay, big and small irregular dots. Cannot see. I think white color is the best. Okay. Okay, so big and small irregular dots. So this is called mosaic pattern, right? But when you're asked to describe what is mosaic, don't tell me about the pattern, because look at my definition. I didn't talk about the pattern. I said protein molecules are scattered. That's the answer you need to write to score. Okay? So you can imagine it, this like icebergs that are floating in the sea, right? Scattered all around, big iceberg, small iceberg, okay? Tall, short, whatever, scattered in the sea. Okay? So that is fluid mosaic model of membrane. Now, the next thing we're going to look at is cholesterol. Cholesterol plays a very important role to maintain membrane fluidity and stability. So remember just now I said that membrane cannot be too fluid, nor can it be too liquid. What am I saying? Sorry. Cannot be too fluid or too viscous. Okay. So we use cholesterol to maintain the fluidity of membrane. So therefore, membrane becomes stable at a wide range of temperature. It will not be too fluid when it's hot. It will not be frozen when it's cold. So this is called membrane stability. So let us look at the structure of the cholesterol. Now pay attention to the structure of cholesterol here. Okay, just pay attention to it. You can see that it's made up of multiple organic rings here. This is the basic structure, multiple organic rings, and they've got a hydrocarbon tail. And then very interestingly, there is something here that is water soluble. Can you see here? OH. Remember OH? For OH, the H is delta positive and the O is delta negative. So it's definitely soluble in water because it can form hydrogen bond with water, right? Okay. So even though we say that cholesterol is lipid, but that this part of the lipid is soluble in water, which is called the OH region. Okay, so it's represented by this part here. So this cholesterol has got a hydrophilic head as well. Just like the phospholipid molecule is a lipid, but it's got a hydrophilic head, right? So therefore, when you throw this cholesterol into the phospholipid bilayer of the membrane, now it will arrange itself in a way that water living part will be facing water and all other water-loving part of the membrane. Water-hating part will try to face away from the water, and also it will facing all the water-hating part of the membrane. So what does that mean? So you see here, which region of the phospholipid molecule is hydrophilic? You see, this entire region is hydrophilic. Okay, and this entire region is hydrophilic. So can you see that the phosphate head of the phospholipid molecule, as well as the OH part of the cholesterol, they are all aligned in this location, right? Because they are hydrophilic. Hydrophilic and hydrophilic will mix. So it will mix with water, it will mix with each other. So therefore, they'll be facing water over here, right? They'll be facing water over here. And they will also be facing each other over here. Can you see that? Okay. And the hydrophobic part will be facing away from the water. So the hydrophobic part is here. All the hydrophobic part of all the molecules in the membrane will be located in this region, you name it. So they'll be facing each other, either fatty acid tail with fatty acid tail, or fatty acid tail with the cholesterol. Okay, so there'll be hydrophobic interaction with each other and with one another, right? So when you draw the cholesterol in the phospholipid bilayer, you use this to draw, you draw it this way, you draw it a rod shape, okay, rod shape, slightly round at two ends there. And you can see that this structure is actually smaller than the phospholipid molecule. You need to pay attention to the size, because your drawing must be quite accurately representing the size. Can you see the size of the cholesterol versus the size of one phospholipid molecule is like 1/3 of it, right? Can you see that? So therefore, when you're asked to draw the membrane structure, you need to draw it accurately. So the size is only 1/3, and the location also must be correct. The location of the head and the tail must be correct. Okay. So function of cholesterol is to regulate fluidity of the membrane in order to prevent it to freeze at low temperature. So you see, when you throw the cholesterol into the membrane, you look at what happens. It actually disrupts the close packing of the fatty acid tail, right? You see here, when these two fatty acid tails are aligned with each other, they can form maximum hydrophobic interactions, right? But when you throw the cholesterol into the phospholipid bilayer, you can see that cholesterol's structure is very interesting one, you see or not? Okay, certain part is facing the left side, certain part is facing the right side, you can see or not? So it cannot, the entire length of it will not form hydrophobic interaction with one fatty acid tail alone. Okay, so you see here, this part can form hydrophobic interaction, only this part, and this part only. Okay, whereas this part here can form hydrophobic interaction with another fatty acid tail. Okay, so therefore it will try to space out the neighboring fatty acid tail to reduce the hydrophobic interaction, but it will still be holding the fatty acid tail together, okay, at certain location. So it prevents the entire length of the fatty acid tail from forming hydrophobic interaction, but it will still hold the fatty acid tails at a certain location. Let me see. It will still hold it at certain location so that it will not be too free to move until it becomes too liquid, but it will not form too much of hydrophobic interaction until it becomes very viscous and freeze at low temperature. So with that property, with that structure, the cholesterol is able to hold the phospholipid molecules in a way to actually regulate fluidity of the membrane. So the membrane will not be too fluid, nor will it be too viscous. So that maintains the stability of the membrane at a wide range of temperature. Okay. So second function of the cholesterol is it acts as a plug. Now, what do you mean by a plug? You know, you go to the toilet and you look at the sink, right? When you open the tap and then the water will flow, it will actually be drained out from the sink from the hole at the bottom, right? So if you don't want the water to drain out, you want to actually fill up your sink with water, you have to put a rubber plug, okay, rubber plug. So in the same way, the cholesterol also will act as a plug because usually what happens is that in between phospholipid molecule, right? If I draw another phospholipid molecule here, right? And they're supposed to have spacing, and then I draw the tail here. Okay, I draw the two tails here. Okay, what happened is that there is a space in between the phospholipid molecules. There's a gap. Can you see here? There's a gap here. There's this gap here. So this gap can allow water molecules to pass through. Remember, water molecules are so tiny that they can actually pass through the membrane, through the spaces in between the phospholipid molecule. So now if I plug all the spaces between the phospholipid molecules, my water molecule can no longer pass through the membrane already. So now this membrane becomes impermeable to water. Okay, so in our body, there are certain cells that have to be totally impermeable to water due to the special functioning of that organ. Okay, so therefore, for those kind of cells, how is it the membrane can be impermeable to water? It means every spacing between the phospholipid molecules will be filled by cholesterol. So it is said that normally in our cells, 50% of the molecule is phospholipid and another 50% is cholesterol. Okay, in plant, there will be less cholesterol, and in bacteria, there is no cholesterol. They don't have cholesterol in the membrane. Okay, they use another molecule which takes a function similar to cholesterol, but they do not use cholesterol. Okay, so do you see that cholesterol actually plays a very important role in our body? Okay, so why is it we say that, oh, cholesterol no good, cholesterol no good? Cholesterol is very important in the body, all right? And every one of us have got our own average level of cholesterol. Different people could be slightly different. So as long as your cholesterol level is in a normal range and it does not fluctuate to become too higher and higher and higher every year, then to me it's totally fine. Okay, it's totally fine. Yeah. Let's not talk about that. Okay, so let's answer this question. The diagram shows part of a cell surface membrane. Past year MCQ. Which components help to maintain the fluidity of the membrane? There are, see, you got two molecules there. Megan?
Is it 3 and 5?
3 and 5. 3 is the fatty acid tail, correct? Because the hydrophobic interaction of the fatty acid tail, okay, can affect membrane fluidity, right? Whether your tail has got double bond or single bond, whether your tail is long or short. Longer means got more hydrophobic interaction. Therefore, more hydrophobic interaction means less easy to move, so more viscous. Correct. Second one is the cholesterol. So where's my cholesterol here? Okay, this is a cholesterol. So answer is 3 and 5. Correct. Answer is D. Okay, correct. Okay, so this is quite easy provided you know your basic lah. All right. Next question. Okay, let's continue. Now we describe membrane as partially and selectively permeable. It is not acceptable if you say membrane is semi-permeable. This is considered as wrong. Okay, never, ever, ever, ever use this term semi-permeable. Because when you use the word semi means half. Afterwards, I'll show you why is it half is wrong. Is membrane half permeable and half not permeable? Definitely is wrong. Okay, membrane is not half permeable and half impermeable. That means to say, I've got 10 molecules. Out of 10 molecules, 5 can go in, 5 cannot go in. How can it be, right? It depends on what molecules you have, isn't it? Okay, so therefore this statement is totally not correct. Never ever use this term again from A levels onward. We always learn to use the most accurate term to describe things. Okay, so why membrane is partially and selectively permeable? So movement of molecules or ions across the membrane depends on the molecular size and polarity. So what I show you here on the diagram on the bottom right hand side is the phospholipid bilayer, and there are three particles that I want them to move across the membrane, the three particles. Okay, whether they can move across or not depends on certain properties of these particles. Number one, small and uncharged molecules, it will be permeable. So like water molecule, oxygen, CO2 gas, ammonia, small hydrocarbon like CCl4, ethanol, all of them are tiny. So they are able to pass through the membrane. So the membrane is totally permeable to them. Okay. Number two, large polar molecules. Large polar molecules got restricted flow, like glucose, amino acid, fatty acid, glycerol. They are larger than the water, oxygen, so on. So what happened is that it's not easy for them to go through the membrane. They want to go through can. They must go through the spacing between the phospholipid bilayer. They must go through the spacing between the phospholipid molecules. And the spacing is so small that most of the time they are unable to pass through. So that's why it said restricted flow. We didn't say it cannot pass through. It's restricted to pass through due to the spacing between the phospholipid molecules that are so tiny. So they cannot pass through because they're too big. Okay, even though they are not charged, but they're too big, they cannot pass through. So third case, ions. Ions also restricted flow, not because of their size, but because of their charge. Okay, don't forget this region of phospholipid molecules here, they don't like water one. Okay, so anything that's hydrophilic comes to here, they push it away. So therefore, my ions cannot pass through the membrane. Cannot, because it's charged. It cannot pass through the hydrophobic region of the membrane. So sodium, potassium chloride, they're very small, but they cannot pass through. So therefore, whether a particle can pass through the membrane depends on what? First, the molecular size, and also the polarity. That means whether they are charged or they are not charged. Okay. So for those molecules that are important for our cell, like glucose, which is needed for aerobic respiration, right? Amino acids needed to make protein, fatty acid and glycerols, all are very important. So if they cannot enter our cell, we need special assistant to help us to bring them through the membrane into the cell. So these special assistants are called the carrier and channel protein. Carrier and channel proteins are used to transport selective large polar molecules and ions across it. So I use the word selective. Okay, that means what they will transport across is very specific. Each carrier protein will only carry one type of molecule. Remember, protein is very specific, so they function based on their shape. Only those molecules that are complementary to their binding site can sit in and be carried across. So we're going to look at that one afterwards, okay? So membrane is a phospholipid bilayer. So in this diagram, it shows you that the membrane is being frozen. Okay, it's being frozen already. And then a small needle is being used to separate the upper and lower layer of the phospholipid bilayer to show the distribution of the proteins in this membrane. So from this experiment, it's shown that the distribution of the proteins in the membrane is not symmetrical for the upper and the lower layer. It is not symmetrical. Okay, sometimes the upper layer got protein, the lower layer don't have. Sometimes lower layer got, upper layer don't have. Sometimes upper lower also got. Okay, so this showing the distribution of protein is not symmetrical between upper and lower layer. So this technique is called freeze fracturing technique. You freeze the membrane and you break the membrane, so fracture. Okay, reveals the arrangement of protein molecules in the phospholipid bilayer. So it is asymmetrical. Okay, so let us look at the protein. We can divide the protein into two types, either intrinsic or extrinsic. Intrinsic means found inside the phospholipid bilayer. Extrinsic means found outside the phospholipid bilayer, but attached to the phospholipid bilayer. Okay. So let us look at the intrinsic protein. Intrinsic protein can be further divided into two types. One is called the partially transmembrane protein, and second is called the transmembrane protein. So you need to draw this into your notes, okay? Partially transmembrane protein, you can see that this blue color is a protein. It is only located at one side of the phospholipid bilayer, so it's partially transmembrane. Partially means part of. Trans means across. Across the bilayer is only part of the across, okay? Transmembrane protein means entirely protein is across the entire membrane phospholipid bilayer, okay? Now, I want you to pay attention to the shading of this protein. Why is it this protein remains inside the membrane in this manner? Is because, again, of the hydrophobic and hydrophilic part of the protein, right? So the shaded part in blue color are all the hydrophilic part of protein. So it will be facing the water, okay? And it will also be facing the hydrophilic head of the phospholipid molecule. Then this part of the protein is hydrophobic. That's why it's located at the hydrophobic region of the membrane, right? It's facing the fatty acid tail, which is hydrophobic. So this kind of interactions will cause the protein to remain forever inside the membrane in this manner, okay? Because of the hydrophobic interactions as well as the hydrophilic interactions. So the question here is, why do the protein molecules remain inside the membrane? So if I give you this as a question, 3 marks, are you able to get the 3 points to score the full marks? You can, right? Okay. So membrane structure is maintained by, so now you can see cholesterol inside here. Can you see the cholesterol? There's cholesterol and also the protein molecules and something that looks like antenna, which you have not learned yet. The rest you learned already, okay? So this is called the entire membrane structure made up of the phospholipid molecules. Let me show you. Okay, the phospholipid molecules. Okay, the protein, right? And then all these are proteins, and then the cholesterol here, right? Cholesterol. Okay, and then of course these fiber-like structure are the carbohydrate chains, right? Carbohydrate chains. So this form the entire structure of the membrane. So membrane structure is maintained by what? All the interactions, hydrophilic interactions between, okay, see, these are all the hydrophilic region of the membrane, right? Whether it's protein, whether it's cholesterol, whether it's phospholipid, all this location here, the molecule at this location will have the hydrophilic part, okay? These are all the hydrophilic part of the molecules. So it will form hydrophilic phosphate heads will be facing water. Hydrophilic regions of proteins will be facing water, and they will also be facing each other, okay? Hydrophobic interactions, right? Hydrophobic interactions is fatty acid tail with fatty acid tail in this region here. Fatty acid tails with hydrophobic region of cholesterol and hydrophobic regions of protein. So these kind of interactions maintain the structure of the membrane. Okay, maintain the structure of the membrane, which is different from maintain the stability is different. Maintain fluidity is different. This is maintain the structure. Okay, so pay attention. What maintains structure? What maintains viscosity? Okay, what maintains stability? All are different. Okay, so pay attention to the details. Right, so let's look at the function. So we look at the arrangement of the molecules in the membrane. Now we want to look at the function of those molecules in the membrane. Function number 1, transport. So you need to draw this up. Carrier proteins, right? Carrier proteins are used to carry large polar molecules across the membrane by changing their shape between ping and pong. Okay, so if this is ping, this is pong. Now, this is the binding site of the carrier protein facing outside the membrane, and this is binding site facing inside. So when the molecule that it wants to transport sits into the binding site, for example, glucose, so the glucose can sit in here because glucose has got complementary shape with the binding site of this carrier protein. Now, I'm not saying active site, only enzyme got active site. Other than enzyme, other protein molecules that function based on the shape, this location is called binding site. It's no longer active site, okay? Because it does not catalyze biochemical reactions. Only enzyme can catalyze, so it's called active site. Other than enzyme, any other protein you talk about is called binding site already, okay? So again, principle is by complementary base pairing. The moment glucose sits into the binding site, the carrier protein will have to hold the glucose, so it will change shape. So remember, this is what property of the enzyme? Allosteric property of enzyme. That means it will take a different shape. So it will change shape. So when it changes shape, this is how it looks like. Look at the animation. Okay, something sits in, it changes shape. That's why it changes shape from ping to pong, ping to pong. Can you see that? Okay, it doesn't turn. Okay, it doesn't turn this way. Okay, it's like your hand open like this, and then something sits in. Okay, something sits in, and then you open the other side and let it come out. See, this one sits in here, and then I close, I open the other side. Okay, I don't turn my hand this way. Okay, no turning. Right, open this side, close, open that side. Okay, this is a fastest way, no turning, right? But changing shape. So this is how carrier protein can carry all the useful molecules into our cell. Now, what about channel proteins? Channel is like a little stream or a little river or a little drain across the membrane. Now, you know that the center part of membrane is hydrophobic, right? But we create a little drain which is filled up with water across the membrane so that water-soluble ions can pass through the hydrophobic part of the membrane. Okay, so this is what you need to draw. So we use a few proteins to arrange in such a way that right in the center of the protein is the channel that is filled with water. So water-filled polar channel. So now my ions can pass through the channel protein. Okay, so it is to facilitate. Facilitate means to help, to facilitate the movement of ions and small polar molecules across the membrane. Now again, different channel proteins will be specific to carry different ions, same as carrier protein. They are very specific. Okay, they only carry one type of ion that's specific for them to carry only. Okay, so every cell is selective of what they want. So therefore, they only allow what they want to enter the cell. So that's how they become selective. Okay. So on the right side, the diagram shows you there are about 2, 4, 6, 8. There are 8 protein molecules arranged around each other to form a center water-filled channel. So this is called the channel protein. Okay. If you look from the top, this is how it looks like. Right in the center, there's channel. At the side are all the protein molecules forming the channel protein. Okay. So this is how ions can pass through the channel proteins, looking at the animation. Okay. Now, next, function of protein molecules in the membrane, cell signaling. So there are some proteins whereby it is used as a receptor molecule. So this protein is called a receptor molecule. Receptor means to receive something, right? So what is it you want to receive? You want to receive a signal for your cell. Say, for example, you want your liver to start to take up excess glucose from your blood and convert to glycogen. So it has to receive a signal to inform itself that, hey, you know that glucose is too high in the blood, please take up the glucose and convert to glycogen. So it needs this signal. So you need a receptor to receive this signal molecule, which is insulin, so that the liver cell now will know that, okay, I'm supposed to do this now. So this is called a receptor function in cell signaling. Okay, so this is called cell surface receptor. So the chemical signal, which is a chemical molecule, which acts as a signal to inform the cell to do something. This chemical is called a ligand because ligand is a chemical that can bind to. So it can bind to the binding site of the receptor molecule. So it binds to the binding site of the cell surface receptor and then inform the cell to do something. Okay, so for example, the signal molecule can be hormone like insulin, and the target cell is like liver cell. Okay, it can also be, this molecule can also be a neurotransmitter, the signal molecule, which is sent from one nerve cell to the next nerve cell. Okay, and this is to activate reactions in the cell. Okay, other functions of the protein molecules in the membrane, they can act as enzymes, right? Like, you know, the thylakoid membrane of chloroplast got a lot of enzymes that are used for photosynthesis. And the inner membrane of mitochondria, remember you said they carry out aerobic respiration, right? So we contain membrane, we contain enzymes for aerobic respiration in the inner membrane of mitochondria. And also the microvilli of our gut cells, they contain enzymes to digest disaccharides. Okay, so that means as the disaccharides are taken into the cell, it will be digested from disaccharides to monosaccharides. Yep. And another function is electron transport chain. Now, can you see these blue color protein molecules are arranged in a series, side by side like this? Okay, so we call this series of protein as the electron transport chain. So it's a chain of electron transport molecules. It's a chain of protein which transport electrons. Can you see the electrons is being transported? So this protein will carry electron and pass to the next protein, and the next protein receive it and pass to the next protein. So this is called the electron transport chain. It's found in thylakoid membrane of chloroplast, inner membrane of mitochondria. They play very, very important role in aerobic, in photosynthesis and aerobic respiration. Okay, now these are carbohydrate chains found in the membrane, either attached to the protein, which we call them as glycoprotein, okay, or attached to the lipids, which we call them glycolipids. Right, so this one is attached to the protein, and this one is attached to the phospholipid molecule. So you got glycoprotein and glycolipid. Both of their function according to your new textbook. Okay, I've modified my notes already because, no, I've modified my PowerPoint, not my notes, because your new textbook only wants you to learn this. Function, first, the carbohydrate chains is facing outside of the membrane. So you know membrane, you got the surface facing outside of the cell and the surface facing inside of the cell. This carbohydrate is found facing outside of the cell. Okay. Function, receptor site for chemical signaling, which I've mentioned just now. Okay, for chemical signaling. Then it also acts as antigen. Now, antigen is the name tag of the cell. Our body got a lot of cells, right? How do you know this? Liver cell, fat cell, connective tissue cell, skin cell, epithelial cell. Okay, so you ciliated epithelial cell. So all cells got their own antigen. So antigen is very important for cell recognition during growth and development, so that they know that, okay, we are supposed to form skin cells, so we stick together, or we're supposed to form the fat tissue, so we stick together. We're supposed to form a connective tissue, so we stick together in this layer only and not other layer. Okay, So it's important for the cell to recognize each other using the name tag of the cell. Okay. Immune response also very important because we want our immune system to only attack foreign cells. Foreign cell got their own antigen, which is called non-self antigen. Our own cell also got their own antigen, the name tag, which is called self-antigen. So we recognize this cell is a foreign cell by looking at their non-self antigen. Then we attack them because they're not our kind, right? They're foreign, okay? And finally, stabilized membrane structure. Carbohydrate chain can form hydrogen bond with water molecules, and therefore it allows the membrane structure to be maintained. Okay, hydrophilic part will be facing the water, hydrophobic part will still be facing inside. So the carbohydrate chain has got a lot of sugar unit with a lot of OH. The OH can form the hydrogen bond with water, so holding the membrane within the water. Okay, so maintain the overall 3D structure of the membrane. Okay, so that's all for today. You have looked at the molecules that make up the membrane and where they're arranged and their function. You have looked at the fluid mosaic model of the membrane as well. So that's all for today. Okay, so please make sure you do all these drawings into your notes. All right. Okay, so that's all for today and have a nice day. Bye-bye, everyone.